Hello. Today
Hello, guys.
we will talk about genetic codes, mutation and translation. Overview of translation. The translation is the second stage in gene expression. Translating the nucleotide sequence of the mRNA or messenger RNA into the amino acid sequence of a protein. Okay? The genetic code is defined as the relationship between the sequence of nucleotides in DNA or its RNA transcripts and the sequence of amino acids in a protein. Each amino acid is specified by one or more nucleotide triplets or codons in the DNA. mRNA acts as a working copy of the gene in which the codon for each amino acid in the protein have been transcribed from DNA to messenger RNA. Transfer RNA serve as adapter molecules that couples of codons in messenger RNA with the amino acids they each specify. Translation takes place on ribosomes. Ribosomes are complexes of protein and ribosomic RNA or rRNA that serves as a machines coordinating in the interactions between messenger RNA, transfer RNA, the enzymes, and the protein factors. Remember always the important features in genetic codes. Each codon consists of 3 bases or triplet bases. There are 64 codons. They are all written in the 5' to 3' direction. 61 codes for amino acids. The other 3 UAA, UGA, and UAG are stop codons or non-face codons that terminate the translation. There is one start codon or initiating codon, AUG, which coding for methionine. Protein synthesis begins with methionine in eukaryotes and formylmethionine in prokaryotes. The codon is unambiguous, or it means each codon specifies no more than one amino acid. And the codon is degenerate, or it means more than one codon can specify a single amino acid. All amino acids except methionine and tryptophan have more than one codon. Okay? Methionine and tryptophan have just one codon. For those amino acids having more than one codon, the first 2 bases in the codon are usually the same. The base in the third position of varies. The code is universal for all. Live life is the same codon. The code is commaless or contiguous. There are no spacers or commas between codons on a messenger RNA. Neighboring codons on a message are non-overlapping. You can see in this table phenylalanine UUC, both code for phenylalanine. Important to remember AUG for start codon or for methionine, and UAA, UAG, UGA to stop codons. The other one is impossible to remember all. So, what means mutation? Mutation means a permanent, heritable change in the DNA-based sequences of an organism. This altered DNA sequence can be reflected by changes in the base sequences of a messenger RNA and sometimes by changes in the base sequences of an amino acid sequence of a protein. Mutation can or not cause genetic diseases. Also can cause changes in enzyme activities, nutritional requirements, antibiotic susceptibility, morphology, antigenicity, and many other properties of the cell. The single base alteration or point mutation can be transcription, transition, or transversion. In transition is a point mutation that replaces a purine pyrimidine base pair with a different purine pyrimidine pair. For example, a T base pair becomes a GC base pair. Transversion is a point mutation that replaces a purine pyrimidine base pair with a pyrimidine purine base pair. For example, a T pair becomes a TA pair or CG base pair. Okay? Or other example, a CG base pair becomes an AT base pair or GC base pair. The classification of mutation, one classification is according to the effect that the mutation have in the structure of the gene's protein product. The first which we'll talk is about silent mutation, or new codon specify the same amino acid. The effect on protein is known. The protein will be the same, exactly the same. In sense mutation, a new code specifies different amino acids, possible decreasing function, and this change can be variable in effects. In a nonsense mutation, a new codon is a stop codon, or the new protein is shorter than normal and usually non-functional. Some shift in frame mutation means the addition or deletion of bases, or usually non-functional, often shorter than normal, because you have an stop codon. Large segment deletion, or unical crossover in meiosis, result in loss of function. The protein is shorter than normal or entirely missing. 5' splice site donor or 3' splice site acceptor result in effects ranging from addition or deletion of a few amino acids to deletion of an entire exon. Expansion encoding region called large segment deletion. Explain now better. The large segment of the DNA can be deleted from a chromosome during a unical crossover in meiosis. Crossover or recombination between homologous chromosomes is a normal part of meiosis, one that generates genetic diversity in reproductive cells, in eggs and sperms. A large beneficial result. The resultant chromosomes are mosaics of maternal and paternal alleles. The example of large segment deletion include alpha-thalassemia, Trinucleotide. Alpha-thalassemia unical crossovers has deleted one or more alpha-globin genes from chromosome 16, and Trinucleotide result in mental retardation, microcephaly, wide-set eyes, and a characteristic kitten-like cry results from a terminal deletion of the short arm of chromosome 5. And the other, which we talked, is mutation in splice sites. They affect the accuracy of intron removal from heterogeneous nuclear RNA during post-transcriptional process. If a splice site is lost through mutation, spliceosome may delete nucleotides from adjacent exon, leaving nucleotides of intron in the processed messenger RNA. Use the next normal upstream or downstream splice site, deleting an exon from the processed messenger RNA. Mutation in splice sites examples: beta-thalassemia, Gaucher disease, and Tay-Sachs. Beta-thalassemia. There are two genes for beta chain of hemoglobin. There is a deficiency of beta-globin protein compared with a globin. A large number of mutations have been described. Gene deletion mutations that slow the transcriptional process, and translational defects involve nonsense and framework mutation. Other mutations involve beta-globin messenger RNA processing. More than 70% of beta-globin gene is not encoding information and eventually must be spliced out, such as splice site mutation at the consensus sequence. Also, mutation within intron 1 create a new splice site, resulting in an abnormally longer messenger RNA. Alpha-thalassemia. A 9-month-old infant of Greek descent was brought to the hospital by his parents because he became pale, listless, and frequently irritable. The attending physician noted that the spleen was enlarged and that the infant was severely anemic. His face had rat-like features due to deformities in the skull. Beta-thalassemia are found primarily in Mediterranean areas. It is believed that similar sickle cell anemia and glucose 6-phosphate dehydrogenase deficiency, the abnormality of red blood cells in beta-thalassemia may protect against malaria. Splenomegaly is due to the role of the spleen in clearing damaged red cells from the bloodstream. Trinucleotide repeat expansion includes Huntington disease, Fragile X syndrome, myotonic dystrophy. The trinucleotide repeat expansion differ from their normal counterparts only in the number of tandem copies of a trinucleotide. The expansion of a trinucleotide repeats in the mutant allele can be in a coding region, Huntington and spinobulbar muscular atrophy, or in untranslated region of the gene Fragile X and myotonic dystrophy. The number of repeats often increases with successive generation and correlates with increasing severity and decreasing age of onset, a phenomenon called anticipation. Normal Huntington alleles have a 5 tandem repeats of CAG repeats. Affected family members may have 30 to 6 of these CAG repeats. Normal protein contains 5 adjacent glutamine residues. The protein code in the disease have 30 or more adjacent glutamines. The long glutamine tract makes the protein extremely unstable. The amino acid activation and codon translation by transfer RNA.